So, hello everyone, it's my great pleasure to introduce Michelle Watt to you, and this is a very special event today because it's actually Michelle's more or less last day at work here in Bonn. She will be moving back to Australia to move to, as a professor to the University of Melbourne, and will continue her career again in Australia where she has been before until something like 5 years ago when she moved from CSIRO to Jülich and Bonn. And it was a great time to have her here. And the last time we have been standing on stage like this was actually 2 years ago when we were the two persons standing in front of the reviewers at the defense presentation for the cluster PhenoRob, and Michelle and me were the ones who had to take the questions from the reviewers and then either answering ourselves or redirecting them to the other PIs who had been around. And it was a great pleasure to do that together with you. I think we have been a really good team. And just by looking to you, we knew already who should answer the question. It was kind of a very perfect show, and I was very proud to be able to do that with you, and we all have been very happy that this turned in such a big success, and that's the reason why you're actually all sitting here today. So this is, you did a lot for that, and thank you very much for defending this endeavor with us. And again, I think thank you very much for the great time you spent here with us. We're really looking forward to your talk, which shouldn't be too much backward looking, just a bit, and then looking a bit more into the future. We are also trying to set up a collaboration with Melbourne. A new training group is in the process of being set up, or at least we try to do that, to kind of keep a tight connection with you, with the other guys from Melbourne, so that you will hopefully stay in the PhenoRob surrounding for the next, let's say, 13 years.
Yes, exactly.
So, thank you very much, Michelle. The stage is yours.
Thank you so much. Is this on? It was on. Okay. All right. Okay. Okay. Thank you so much, Cyril. Yeah, I'm actually really overwhelmed by just the really wonderful things people have been saying about the last 5 years of my time here in Jülich and Bonn. And I have to say that the PhenoRob cluster was just a completely unexpected bonus that came out of the region, and I think not only did it gel a new community that I really think will be very long lasting, but to me it really stimulated and gelled very important scientific discoveries that need to be made for PhenoRob, and that's actually what I'm going to focus on today. It's in the area of dynamics, and I have a focus, of course, below ground, which is the main area that I've focused my career on roots and rhizospheres, the interaction with soil, but these dynamic opportunities that we have now with PhenoRob open big questions across biological systems for us. So yeah, I've always worked below ground or down under or whatever you want to call it, and the region the word that I'm going to use for this region of roots and their interaction with soil is the term rhizosphere. Rhizosphere was a term that was introduced. It really means the soil influenced by the presence of roots, particularly from a biochemical point of view. And it has received a little bit of an artificial division between the root itself and this soil that's associated with the root. And you can see all this root, the white stuff is root, and the soil in this image here. We just published a paper in Trends in Plant Science where we really urged the community to treat the rhizosphere as the root and the soil as an integral entity. And in these two images, this is the rhizosphere phenotype, what it looks like, what's in it, the features of it at one point in time. This is the whole root system. This is a cross-section through a root. And basically, the roots and this rhizosphere zone are responsible for the capture of all resources for plants except for carbon. So water, nutrients, signals, compounds from the soil, are all coming from this zone. And the leaves, respect to Uwe and his team and Ono, of course are important for photosynthesis, but for every carbon fixed in photosynthesis, at least 400 water molecules need to be captured from the roots to fuel that photosynthesis. Now, just see if I can, yeah. So now imagine that those images that I just showed you, that rhizosphere phenotype at one point in time, as it's now having to function over time to support this growing wheat crop. Just imagine the dynamics that are going on as that root system itself grows into the soil to at least 2 meters deep, usually, probably much deeper here in winter wheats in the northern hemisphere. And it is constantly supplying water and nutrients for this leaf growth. It is changing the soil environment that it's coming in contact with, and therefore thousands, well, every millimeter of root has 1 million bacteria associated with it, and these are changing constantly through time. And then finally, as the crop reaches yield stage, it is finally enabling those last resources to go into the grain so it's harvested. And really, we have a very, very interesting scientific opportunity now, which is around understanding the dynamics of biological systems, and in this case, from the below-ground perspective. This is not an insignificant statement. Plant biology really still is really using concepts that are bottom-up, gene to phenotype. They are quite simplistic in terms of the conditions that plants are studied in. And then finally, they are dealt with from a static point of view. And we really need a new conceptual framework in biology to underpin the fact that they are dynamic. So why should we understand these dynamics, particularly from the context of agriculture? Well, focusing particularly on inputs of synthetic fertilizers. This is mainly nitrogen, but it's other fertilizers that are mined, such as phosphorus, micronutrients, etc., etc. And in this graph here, these are all countries across the world where the amount of fertilizer that they're applying per kilogram of hectare is plotted against the cereal yield per kilogram per hectare. And it is linear. And essentially, it just means that agriculture today, for the most part, relies on agricultural inputs of synthetic fertilizers. There are some outliers who, for example, here, these countries here, well, Jordan, for example, interesting, high amount of fertilizer to a relatively low yield output. And then there are, for example, some quite efficient systems. Let me see if I can actually find Australia. Yeah, Australia is right here in the middle. So it actually, because its farmers don't receive subsidies for inputs in agriculture, they have a relatively good efficiency of cereal output per kilogram of fertilizer. But all in all, globally, this is linear. Now, why should we worry about cereals? Well, this is the agriculture land used by major crops globally in total. This is the cereals, and this is everything else. So the cereals, wheat, maize, rice, rye, grasses that are fed to animals, cereals, they are the main game in town when it comes to thinking about reducing inputs. This will have a big global impact by focusing on cereals. Now, what's happened over the last 2 or 3 years is that there has been an increasing expectation that biological seed treatments, so microorganisms applied to seeds, sold to the farmer, are going to be able to actually carry out biological mechanisms with soil to provide fertilizer from the soil to allow farmers to reduce their input of these synthetic fertilizers. And this is actually, in some ways, sort of the real deal. This is a global market forecast for the price expectation growth in companies that are investing in seed treatments, so microorganisms that can be applied with seeds. And this is growing very rapidly. And this is why very big companies and small companies have been bought by big companies to incorporate microorganisms with seeds. And this has received a lot of interest for a long time, since the 1980s, as a potential way of reducing inputs. You add microorganisms to the seed that then colonize the root. They carry out biological mechanisms to help the roots get extra nutrients from the soil. And so far, it basically has been not particularly successful. And this recent paper in PNAS just gives an idea. They did a field experiment. This is conventional current agriculture with normal fertilizer inputs for maize and wheat. This is a reduced fertilizer input, and this is an entirely organic system, so relying on the microorganisms that are in the soil. So to add microorganisms and expect gains, there's a big gap that needs to be addressed. And so I'm going to give 3 examples of studies that I carried out in Jülich over the last few years. I was attracted to Jülich, I must say, primarily because of their non-invasive phenotyping technologies and the potential to look at dynamics of the systems. And I'm actually going to start with a field example of dynamics that could be readily exploited in PhenoRob. So in this image here, in this example, rhizospheres for sustainable intensification of wheat, what I'm going to talk about is basically what precision placement of seeds and other inputs, what PhenoRob is based around, allows the farmer to do with increasing accuracy is to place the new season crop, this green line here, very strategically in relation to the last season's crop. So this is a field of wheat where the new wheat crop has been sown in between the rows of the dead wheat crop. This is a fully sustainable no-till stubble retention system. So this is the type of system that farmers are encouraged to go for globally. So they don't till the soil. They have last year's crop rows right here, and then the next year's crop rows can be placed wherever they want because of the precision sowing equipment available today. Now, what's going on there? So as I mentioned, they are not plowed soils. So the soils are highly structured. They tend to have pores and spaces that were occupied by roots. And the new root crop, the roots of the new crop, this root here in white, tends to go into the pores and spaces where the previous dead crop roots were. And we quantified these aspects, and more than 50% of a crop typically contacts a dead root from a previous crop. And we looked at the microbiology of these associations. We followed the first wheat crop and then the second wheat crop, and we harvested the roots and the rhizospheres of the new crops and the dead roots over time. And what was immediately evident from from microscopy studies, this is a new wheat root with a root hair right here. This is a dead wheat root right here. And these roots have been labeled with fluorescence in situ hybridization DNA probes for different bacterial types. And the dead roots are colonized by a different spectrum of microorganisms than the new roots. But when the dead roots sit in places of the new roots, the microorganisms are actually changed and spatially shared in that spot. We went on to do full metagenomics of this succession, this evolution of the rhizosphere, and we were able to identify certain microorganisms that are highly specific to a certain time, and other microorganisms that actually are there for the entire length of time. What we don't know really very much about is the functions of those microorganisms. But what it says from a bigger picture is that in PhenoRob and in other technologies that can place roots of the new root system strategically beside, in, or far away from the next crop root, you can construct the rhizosphere based on the chemistry and the structure of the root systems. The structure and the chemistry of the root system is what is feeding these microorganisms. So you can design your rhizosphere by rotating the roots in space with PhenoRob-based technologies. We have a big scientific question, as I mentioned. We don't really understand what is driving these successions. It would be really good to know why some microorganisms hang in there the whole time. This would be a desirable thing to know for organisms that are being selected for applying onto the field. And also, we really don't know what is driving the whole dynamic evolution of the system. Okay, so I'm going to go on to my second example of rhizosphere dynamics. And this time, this example is in the context of global warming. So we know the situation with the atmospheric warming, which appears to be accelerating even faster than people expected. And the issue that there is in the literature is that very few studies of plant science, when they have looked at the effect of warming on plants to predict how plants will behave in different agroecosystems, is that very few experiments have actually treated the soil and the soil column with the appropriate temperature gradients that they would experience in the field. So often the plants are in a growth chamber that is warmed, but it doesn't actually, many studies, in fact, it's very likely that the below ground information that we have about future warming scenarios so far from the literature is incredibly sparse or possibly incorrect. So what we wanted to do was take a look at the way the rhizospheres were responding to real temperature gradients. And in collaboration with Claudia Knief and Robert Kohler, who's sitting here, we picked two agroecological zones, important ones globally: the German Northern Europe winter wheat zone. This is a map of wheat yields. And we took this central India climatic zone. And Andong measured soil temperatures here in Germany down to depth. I had some previous collaborations in India, and I had soil temperatures from central India. This is actually the field dynamics temperature gradients when wheat is sown in Europe, so quite cold, but dynamics from the top of the soil to deeper soil layers where it's much more flat. And central India, this is surface soil layer, very big dynamics in surface soil, and less with depth, but you can also see much warmer. Wheat is sown between 10 and 30 degrees sowing temperature in the soil in India, and it's sown between 2 and 4, 5, or 6 degrees in the northern European conditions now. So Andong mimicked these conditions in a specialized growth setup. He recreated those gradients exactly with a warming increment of 5 degrees for both of them. And using the MRI system, which lots of you have already seen, he scanned the roots over time, and we also harvested the root tissues, and working with Claudia and her group, doing a full metagenomic study of the rhizosphere microbiome. And what the MRI allows you to do, the magnetic resonance imaging, it scans the roots directly intact in soil. So it allows you to see how they're growing over time and make decision make decisions in terms of harvest points for guided sampling of particular features, in our case, microorganisms. And so we had one harvest which was similar in time, and another harvest which captured similarity in development. And we're just writing up these results now, but I'll summarize them here. Oops. Yeah. Okay, the very interesting thing is that the 5-degree warming had a much, much bigger impact in the German scenario than in the Indian scenario. The German scenario wheat germinated and emerged and grew much more quickly in response to the warming than the India scenario. And we saw a shift in the root architecture. In the German warming scenario, the top soil layer roots were promoted to grow, while there was a reduction in the deeper growth of the roots. And in the Indian scenario, we saw an increase in deeper root growth, but not in the surface roots. So there is a big difference in the way the carbon is input in those two systems in terms of in response to the 5-degree warming. And we also saw shifts in the microorganisms that were tightly and loosely associated with the root in those two scenarios, and as I mentioned, we're writing that up now. So in the final example of the 3 examples of dynamics, I'm going to talk about experiments where we actually have been testing different microorganisms that can be added to the plant for benefits in terms of allowing us to reduce the input of artificial synthetic fertilizers. And we asked the question, can we increase nitrogen capture by cereals by adding biologicals, microorganisms, to the rhizosphere? And this work is being done by a postdoc, Wei-Kui Kwang, and led by senior scientists in the root group Boriana Arsova. And we're working in collaboration with a group in Berkeley, and I'll tell you a little bit about this after I show what we're targeting here. So typically, when cereals perceive that they have sufficient N, so there is nitrogen present there, they behave quite conservatively in the sense they shorten their root systems and they concomitantly increase leaf area. And this is a bit of this shortening of this total root length is a problem because it reduces the colonization length for capturing nitrogen from the profile, particularly in European situations where there's excess and leaching nitrogen. So we wanted to find bacteria that would actually shift this allocation. And what we've been using, and I'm going to continue to use this, is a really neat idea developed by the groups in Berkeley, which is to 3D print lab experimental equipment. And one of the big problems in plant microorganism interactions is it has been very difficult to repeat phenotypes from lab to lab. So they introduced these 3D printed ecosystems, fabricated ecosystems, they call them ecofabs, to allow different labs around the world to repeat and test the same types of experiments. And they can be built by any group, and you can repeatedly collaborate with other groups very, very, let's say, accurately, because they have this exact same system. So we've been making use of these a lot. We adapted them to be able to measure dynamic processes in them, so non-invasive leaf area measurement, non-invasive root length and architecture measurements, non-invasive root hair measurements, and non-invasive microorganism interaction measurements. Highly, highly artificial, but sterile, controlled, and repeatable across international labs. And just to give you a brief bit of information from this study, yes, we actually found one microorganism species that was very interesting. We saw the typical response when the plant receives more nitrogen, it grows a lot more leaf area, but very interesting. So this is low nitrogen, this is high nitrogen, and this is the root length under the 2 nitrogen levels. So the plant shifts the leaf area, but not the root length. But the microorganisms themselves, you see these two bars right here, between here and here. The open circles, no microorganisms. The filled symbols, microorganisms, both at high and low root length, actually stimulated total root length. So it created the phenotype that we were quite interested in. We actually saw a shift in allocation towards more root length than leaf area. And because we measured the dynamics, we were able to look exactly at what part of the root system was shifted, and it was actually only the primary main root that was shifted, increased, at no expense to lateral roots. And also we saw this interesting shift in root hairs, which I won't talk about right now, but we're very interested in how roots actually sense their environment, and the root hairs are the first line of defense. So in summary of those, showing those three research examples, one of the things that we are thinking about conceptually are do plant-associated microorganisms actually interfere with these dynamic allocation processes? And actually, is this a way by which microorganisms influence plant performance? There had been an expectation that microorganisms would cause an effect on the plant, and this would stay there for the whole duration of the crop cycle. It's clearly not true, and it's clearly from these studies showing that they can actually go in and out in terms of interference with plant functions. And we really have, as I mentioned, an opportunity to shift these dynamics with new technologies. And one, of course, really is the phenotype, the PhenoRob concept. And to me, the PhenoRob concept is very much one of glasshouse or lab to field, in this rotating interaction of field information feeding back to controlled environment information, not only to discover new traits. I think we actually have a lot of candidate traits, but really to be able to identify new traits in these dynamics of these allocation patterns. So with that, before I move on to a brief summary of some Australian plans, I really want to thank all my colleagues in ULIC. This is the root group here that we built over the last few years, started when I arrived and built it up. And here's IBG2, many members you've met. Robert, for example, running the MRI facilities, and the Plant Phenotyping Center led by Fabio Fiorani and Kirsten Nagel. Now, these are the important people. This is the PhenoRob Underground, based in ULIC. You have to dig them out and get them over here. There's a very, very strong cohort of junior and mid-career scientists based in ULIC. There's largely this group here across IBG2 and IBG3 are engaged in modeling. And over here is sort of more the experimental people from IBG2, including Josephine Kant and Vera Hecht. And they're working in the soil root theme in Core Project 3, led by Andrea Schneff largely. Now, I want to show a little bit about a project that I think would be very interesting for PhenoRob to keep an eye on. This is a project funded by the International Wheat Yield Potential Partnership. I keep on getting confused with the acronym, but anyway, that's it there. And this is a large international project led by NIAB in the UK. And IBG2 is involved. Uli Schur is replacing me as the lead PI, and Josephine Kant and Vera Hecht are the ones doing the experiments. There are 7 institutes and 5 breeding companies, all focused on basically incorporating root traits into new wheat lines and testing them to see if they actually function in the field to increase yield. And this will go on for the next 3 years. And this is really relying on IBG2 ULIC for the phenotyping. It's a glasshouse to field exercise, and Josephine Kant is the root geneticist. very much involved in the glasshouse phenotyping. She's leading the experiments of the whole project, and Vera is involved in developing field technologies. And this new field coring and imaging platform has just been developed between Bonn and Jülich. Dr. Oliver Schmidtmann, here from the Faculty of Agriculture, has been working very hard with Vera. They have built an automated corer for roots and soil through to 2 meters, and also a portable in-field imaging platform for root length over time. And so this is now available for PhenoRob as well, and both groups are very committed to continuing to automate this phenotyping capability for PhenoRob. Okay, finally, back to the future. I'm going back to Australia. As Cyril mentioned, I came from CSIRO, although it's not quite back to the future because I'm originally from Canada, but that's another story. I was very fortunate, I feel, to get a really excellent position at the University of Melbourne. The University of Melbourne is Australia's #1 university, and actually already has a very strong link with the University of Bonn through the Faculty of Agriculture, Faculty of Medicine, which we're now building with the Faculty of Agriculture. So I will go there and I will start a new group. And the focus of my group initially, what I'm very interested in are the interaction between these plant dynamics and water. This is basically kind of an obvious situation, but there's been surprisingly little fundamental basic plant biology in understanding how plants actually respond to a watering or a rainfall event after a drought period. And so globally, this is recent data, this shows the regions across the world and the percentage of 10-day periods that are in a drought. And they're increasing, and you can see the regions here. But what is interesting about them is they come out of the drought. There's a recovery period because there is water again. And here is some sensor, this is a color map of sensor data of soil moisture, a bit of like what you can generate in PhenoRob. This is a wheat crop season, starting from small plants to deeper, bigger plants. You can see the water capture is getting deeper as the crop is growing, but you have these interesting places where it rains, and then the root systems have to quickly shift in their growth and functions. And we did a collaboration with Amelia Henry in Erie in the Philippines. Marie Klein was a master's student here in Bonn, and we found genetic variation in plants in terms of how they respond to a rainfall event after it rains. They have different genetic expressions of whether they grow the root systems first, whether they grow the shoot systems first, whether they behave conservatively, or whether they behave rapidly. And so I'm very, very interested in these processes and exploiting them. I've actually written one grant. It's a 5-year proposal to the Australian Research Council. It's a bit like the ERC. It's a single-person, large 5-year grant. And I have proposed to work on this intermittent rainfall and water pattern that crops experience, particularly looking at microorganisms that might influence those dynamics to speed up solutions for water and food security. And these people have agreed to be part of the partnership. And then finally, I'll just finish by mentioning to Cyril, Melbourne and myself, we're very committed to seeing whether we can generate an international research training group with the University of Bonn and Melbourne. There are many advantages of proposing such a relationship. One is the 2 agroecological zones that would be covered. There are a number of overlapping targets in terms of reducing inputs. There are some technologies where there are strengths on both sides that can be combined. And I think there are some really interesting visions that are going on, particularly originating from PhenoRob around complex stresses, dynamic solutions, and lab-to-field concepts. And one of the exciting things is just a couple of weeks ago, the Faculty of Agriculture approved the awarding of joint PhDs between Bonn and the University of Melbourne, so we can recognize students. So with that, I'm going to finish. I want to highlight this meeting that's coming up in the US in 2020. You can go on Twitter and see what's going on. And I look forward to seeing you all soon.
Okay, we have time for questions. And I start with one. So when you were showing that if you do the experiments here in Germany and you see if you increase the climate by 5 degrees, how the root growth changes. And I was more surprised that the root growth goes more in the upper part of the soil and doesn't go down, because I, as a non-root person, expected that the roots grow deeper in order to access a water resource which are not that much affected by the increasing climate. So how does it come together? Is this a strategic disadvantage of the plant that we grow here? Or what the explanation?
Interesting.
What's your explanation for that?
Yeah, so our explanation for that is related to the temperature, the way the temperature profile changed. So the German temperature profile on the surface, as you could see, is quite cool. Okay, so when you have the 5-degree warming, because of the shape of that profile and the way warming affects temperature, you get a warming in that top layer, almost of 5 degrees, almost equivalent to the air, but very little change in the deeper temperatures. Okay, and root growth is very sensitive to temperature, and so that warmer temperature seems to have directly stimulated root growth that are physically in that warmer layer. It has very interesting consequences potentially because of the way nutrients are distributed, where water is distributed, and this is one of the things that we're really discussing about the International Training Network is these complex conditions that are in soil, the complex stresses, right? They're all interacting. So the root maybe should go deeper, right? But it's got this other signaling mechanism that says, hey, it's warm up here, sorry, we're going to grow more branch roots, right? So yeah, that's our current explanation for that. While the Indian scenario, it was already hot. So 5 degrees warmer to 30 to 35, it's a no-go zone down there. So it made deeper roots.
Okay.
Yeah.
So we need to heat up the soil deeper below to make the root growth stimulating, going deeper.
You really need to watch out for droughts, because if wheat puts on too much biomass early in the winter months as it warms, it could be problematic later at grain filling. Yeah. Yeah.
Further questions? Mark.
Yes, thanks for your interesting final presentation here in Background.
Thanks.
I have a question regarding your example of the 10-day drought period that you showed, over which you were looking for traits that anticipate better.
Yeah.
Can you comment on the uncertainty that we have on these estimates of the changes in the patterns of the drought?
Yeah.
All sorts of drought, early spring, late summer, late summer drought, and the frequency and occurrence of these droughts is highly uncertain, at least what I know. And I would find it very difficult to look for appropriate traits that are best suited to anticipate this.
Well, yeah, I mean, I really value your thoughts generally on these ideas. We could talk about them later. So I completely agree with you, right? So there's increasing uncertainty about weather patterns and drought periods and when they're occurring. And so my concept with dynamics is that the traits are not fixed traits, which is what the traditional selection programs, let's say the physiology contributing to breeding, has been focusing on. They need to be dynamic. They need to be underpinned by dynamics that can respond to different conditions. And this is this is not particularly popular idea with breeders.
Yeah. Would it also be a strategy to go for diversity, where you have, let's say, a diverse set of genotypes that have different traits?
Ah, sure. I mean, you could plant different genotypes. Yeah, that of course is another option. I still think there is all of this droughting and recovering and constantly happening, and I don't know, we may be wrong. I just feel there's something in those processes that are worth looking at. Yeah.
Okay.
Yeah.
How much is known about what the solar rays are changing in terms of the root zone? I mean, I was sort of reading about plans to cover fields with solar panels so that plants would sort of grow a little bit in the shade. Is that a factor, or is this really a question of temperatures ultimately?
No, it is so the amount of photosynthate, the amount of that the leaves are catching in terms of light and converting to sugars, is another big determinant of root growth. And that is an interesting concept. There are a number of conditions like what Frank is talking about, particularly in Australia, where you actually don't really want that much leaf growth at certain periods. And so some people have been, selecting for different leaf area properties to control that carbon capture and prefer to have it below ground. So maybe you could use shade. That's interesting. Yeah. So light has a big role on root system development. Yeah. Indirectly and directly. Yeah.
With respect to the first project, where you rotate the new crop into the spaces of the old one, did you actually compare yield or any other parameters with respect to whether you have a more homogeneous field array and this striped field array? So, did I understand correctly that this is something that you would promote to have this field array?
Yeah. I mean, this exists in many systems. That array is basically the old crop that is just cut this much from the ground, and all that stubble and everything is left there. And then the farmer can decide whether he puts the new seed actually right onto that row or in between. There are quite a few yield studies done on whether the systems have a higher yield if you plant right on the dead row or on in between. And this all depends so far on major rotations. Like if it's canola and the legume, which is the first crop, it benefits the wheat crop to plant on top of those residues. But if it's wheat on wheat, which was actually the focus of this project, generally the second wheat crop yields lower. And this is actually the objective of this study, was to look at why this second yield is lower than the first. And the feeling there is that in the second wheat crop, you want to plant it away from those dead, partly for major diseases, but we think there's a lot of microorganism interactions there. So that's what's called a no-till stubble retention direct seeded system, which is used in Australia and other places where they've just really trying to reduce their well, it used to be all about maintaining soil to minimize erosion, right? But what's happening is farmers save a lot of money the numbers of times they drive around with their tractor, and this is increasingly going to be a pressure because of the cost and farm sizes. So that's a future system for sure in other places as well.
I have a very practical question as we are just preparing the PhenoRob experiment about root temperature and profiles of root temperature. We have another radar, it's important, we have to monitor this. I just realized we do this according to the standard way with up to 1 meter depth, 4 or 5 different measurement depth, and the recording, I think it's 1 measurement point every 10 minutes.
Yeah.
Is this enough, or do we have to rethink this to get better data from the start on than the standard?
No, this is enough. If you really wanted to, yeah, like for example, if you had different shading arrangements or density arrangements, and you really wanted to see whether that affected particularly the surface layers. So if you've got one, if your first one's at 20 centimeters, right, that 20 centimeters is on the deepish side, because as you can see, this is sort of where the gradients are. But there are very good models that you can put this data into to give you roughly those upper zones. But if you have treatments that are influencing the surface layers in terms of their temperature, density or stubble retention or soil color and stuff, you might want to add another couple of probes in there. Yeah. Yeah.
Further questions?
I have a small question to one of the slides.
Yeah.
Showed the change in the root length because of the manipulation that you made. And you said there is some reallocation of biomass towards the root,
Yeah.
towards the root system.
Yeah.
Where does the biomass come from? Did you see that in the leaf area?
Yes. Yes. So
Reduced leaf area?
you reduce leaf area. Yes.
So reduced leaf area, more roots?
More.
And the net effect on the biomass yield? What is that?
Okay, I should correct myself. So these are seedlings, and we don't have biomass per se. We have leaf area and root length. So because it's non-invasive measurements. So I don't know whether there's a loss in biomass. But what we did do with this experiment, which I didn't show, is we measured the dynamics of nitrogen uptake, including into the tissues. And it does look like there was a 15% contribution of additional N from the microorganisms. So we did a real-time mass balance through time of both N forms and their allocations patterns. So there does seem to be an additional contribution, at least from a nitrogen point of view, that might have stimulated a net overall growth, let's say. But I don't have the biomass data. Yeah, yeah.
Okay. Time for last question. So if this is not the case, then thank